Hello everyone, welcome back to another IB Biology G4 Lab video. Sorry it's been a while, it's been a very busy few months, but today we're back with arguably one of the harder concepts in IB Biology syllabus, the Biochemistry unit. In this video I'll only be covering one of the topics, which is amino acids and proteins, and one day maybe I'll do carbohydrates and lipids. Starting this lesson off, I believe an important thing to consider is you should probably have taken a bit of general biology and general chemistry at this point, or else this lesson will be quite difficult, but it should be easy enough to follow along even if you haven't. And if you have already taken IB chemistry, that is very good. Alright, let's start off with our first topic here, which is on amino acids. Now, the first thing to consider here is what makes chemicals organic. Now, the most common elements in living things is carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. But what truly makes something organic is carbon and hydrogen. Nonetheless, in an amino acid general structure, it contains both carbon, hydrogen, and nitrogen, and one hydroxyl, which is just oxygen and hydrogen bonded together. Oh, there's also an oxygen. Now we can see here that the basic amino acid chemical structure contains a central carbon atom bonded to hydrogen, carbon, nitrogen, and an R group. Now let's break this down into different components. The very left one we have here is the amine group, which I have colored in green. This contains the nitrogen single bonded to two hydrogens. And these are, this is the amine group, and it accepts the protons and comes in handy later when it comes to the protein structures. Then we have a lone hydrogen atom for structure at the top. On the right, we have the carboxyl group, which is a carbon atom that is double bonded to one oxygen and single bonded to a hydroxyl on the side, and this one is negatively charged. Now, the most important component of an amino acid is probably the R group. Now, the R group, as you can see on this chart on the right, can come in various shapes and forms. This is what makes the amino acid unique from one type to another, and it attaches to the backbone of the amino acid, which is what we just covered. The central carbon atom bonded to the nitrogen, the carbon, and the lone hydrogen. The R group is not part of the backbone, because the backbone is the part without the R group. Now, these R groups attach to the backbone and ranges in several shapes, and these different shapes and chemical properties lead the amino acid to react differently to their surroundings, which eventually helps give different functions and protein shape later on that we'll cover. Now, here are some properties of these R groups that you'll see. One of them being hydrophobic. These R groups can be hydrophobic when they're mostly composed of hydrogen and carbon, and these cause it to be nonpolar and hydrophobic, meaning it avoids water and it will cause the protein to fold inwards. On the other end, we have hydrophilic R groups that are charged, and polar will be hydrophilic, meaning it likes water and it will be more favorable towards the aqueous environments and fold outwards towards them. Then we also have the charged R groups, which if you've taken chemistry, you would know that ions of chemicals have charges, and this works just like magnetism, and it can cause the structure to move as a result of these relations between positive and negative. And this way, they can also cause the R groups to lose or bond with hydrogen. And finally, we have the polar R groups. These have partial charges due to the uneven sharing of electrons within the R group, which we'll cover in a different video, but essentially one side has a little more positivity and one side has a little bit of negativity, which causes the R group to be polar and hence more hydrophilic. Now, within the human body, there are 9 amino acids that cannot be synthesized on its own. It needs to be obtained through external sources, and these are called essential amino acids. Now, when they say essential amino acids, they don't mean that the other 11 are not essential. It just means that these are essential to be consumed through other sources. Now, these essential amino acids are mainly found in animal meat food sources that you need to consume, but these are the complete food sources and the complete proteins, and it can be incomplete if other protein sources, such as tofu, will not give all 9 but only some of them. And of course, on the other end there, we have the 11 amino acids that can be synthesized by a regular human body. And nonetheless, again, all 20 of these amino acid types with the R groups are vital for any proper protein function, but the IB won't ask you to memorize what these essential and non-essential ones are. You just need to know of their existence. Then there's the universal code. Every existing species on Earth uses the same universal code here to identify the different amino acid bases, which is U, A, C, and G, which are the nucleotide bases that make up the mRNA codon. Now, if you remember, DNA transcribes information into the RNA, which is the UACG that we have here, and then that eventually translates at the ribosome to become proteins and the amino acid chains, which eventually folds to become an actual functional protein. We're at the stage where it's after been transcribed, and this is the RNA section, and it's written in the UACG, which can be translated into a primary structure. Alright, let's move on to our second topic, which is fittingly peptides. Now first we need to learn about condensation reactions, which are when monomers, such as amino acids, are condensed into becoming polymers, or in this case, peptides. This is an example of an anabolic reaction, which the opposite would be a catabolic reaction of hydrolysis. But for now, this case, we'll be focusing on condensation. So these monomers, to become polymers, they require energy from an outside source, such as ATP, to be inputted into the equation. And together they're able to condense into these polymers with an excess H2O as waste. Now, let's see the amino acid structures that I've put on here on the screen. When you add these two together, you can see that they will happen there next to the hydroxide and the hydrogen. These two fall off and bond together to form the excess H2O that I mentioned earlier. And from those two, the carbon and the nitrogen, which now both have an empty bond to fulfill, bond together to form what is called a peptide bond. Now, this process occurs in the ribosomes. This is where the translation occurs from earlier, and this is how the amino acids are able to become the primary structure of a polypeptide. Now, as stated earlier, there are 20 different R groups that are possible within each amino acid, meaning that in a peptide chain, each amino acid has 20 different possibilities, meaning that for every number of amino acids, it compounds for 20 different possibilities of new variation. So the equation to find the total amount of variation of a peptide chain would be 20 to the power of n, in which n is the number of amino acids. Now, some terms you would want to know, which are quite obvious, is dipeptide, which is a peptide with 2 amino acids, an oligonucleotide, which is 3 to 20 amino acids, and a polypeptide always has 20 or more amino acids. And a useful equation to know is that the number of peptide bonds is always 1 less than the number of amino acids. Alright, let's get into the heaviest topic here now, which is proteins. The protein has 4 different levels of structure, per se. There's the primary, secondary, tertiary, and the quaternary. The primary is the easiest. It's what we just learned. It's the amino acids with peptide bonds holding it together. And the properties of the R groups in this primary structure eventually determine the final structure of the protein when it gets to the higher levels, which I'll get into in a bit. But essentially, this is the polypeptides that we just talked about and are held together by peptide bonds in the amino acid backbone. Secondly, we have the secondary structure, which is more of a repeating pleats and helixes that form as a result of hydrogen bonding in the amino acid backbone. Now, there's only 2 shapes of these that we can find in the secondary structure, which is in most of them. The alpha helix, which is a helix structure that is a result of hydrogen bonds in every 4 amino acids that circle around a central axis that doesn't really exist, but it eventually does form the spiral shape that you can see. And there's also the beta sheets. The beta sheets are held by a chain that folds back on itself and performs hydrogen bonds between the different backbones and creates kind of a folded pleat shape. Then we have the tertiary structure, and this is when the proteins begin to become three-dimensional. And this is mainly structured through the interactions between R groups, and there are main 4 processes in the R group interactions that cause it to have this structure. One of them being the hydrogen bonding between polar R groups, then hydrophobic interactions between nonpolar R groups that fold inwards because it's trying to avoid the aqueous environment around it. There's also the disulfide covalent bonds, which is sulfur ions that try to bond together as a result of the cysteine amino acids in the R groups. Well, the R groups are the sulfide in the R groups, I mean. And then there's obviously the ionic bonds with charged R groups, which bond together through electromagnetism. And finally, we have the quaternary structure. The quaternary structure here is also three-dimensional, but the key difference is that there's more than one polypeptide chain involved. And the same interactions as tertiary groups will still apply here, such as the hydrogen bonding between polar R groups, hydrophobic interaction between nonpolar R groups, disulfide covalent bonds, and the ionic bonds between the charged R groups. Quaternary structures, however, have 2 different types. There's a conjugated and a non-conjugated. The conjugated proteins have an attached chemical group that provides additional functions. And these additional groups are called the prosthetic group. So one example of this would be hemoglobin's iron groups. Outside of just the polypeptide chains, there is an iron group, a prosthetic group that contains iron to be able to bond with oxygen better, to hold that oxygen and release them. On the same end, non-conjugated proteins are the opposite. These proteins are simply made out of the polypeptide chains, and the functions merely come from them. Now, when we get to this final level of protein, we can classify them into 2 different groups, fibrous and globular. The fibrous proteins are, as it sounds, long, thin strands that have repeating structures used for mainly supporting the structures of the protein and the cells. They're very resistant to any changes like temperature and pH and are very structurally strong, and globular is more the opposite. They have low resistance and very low resistance to temperature and pH changes. And they're not really used for structure. They're more spherical and compact with more containing diverse roles, such as enzymes, which can catalyze reactions, hormones for messaging and transporting materials throughout the body, such as hemoglobin carrying oxygen. At this final level, proteins can also be marked through different modifications, such as, let's say, ubiquination, which can add ubiquitin to mark for degradation at the proteasome. Now, this process is mainly that ubiquitin can tag proteins along the way, and this makes them recognizable to the proteasome. And the proteasome here degrades the protein to prevent stuff that is unrequired after a certain time, such as the cyclins after the cycle's done, the clumping, and eventually it breaks them down into protein fragments, which are eventually digested into becoming individual amino acids, which can repeat the entire process that we learned in this video. And that is it for this episode of the Biochemistry Part 1 unit of amino acid and proteins. Thank you for watching, and of course it is almost time for the IB exam season, so it's time to get studying up, and we will try to produce one more video here for you guys before the IB exam, and then we'll probably be passing this project down to the next group and the next year for those who want to continue it. Thank you for watching this video, and next episode we'll probably be covering something along the lines of integral proteins and cell membrane structure. Thank you.